All right, now let's talk about section 2 of chapter 15. We're going to talk about some more high-tech ways of manipulating the genome. So we talked previously about how you can make use of the natural variation out there to introduce new genes into your breeding pool. You can induce mutations sometimes. But now we're going to talk how we can cut out and move specific genes from um, one individual to other individuals, or even between species and between kingdoms, using sort of normal artificial selection techniques. You're restricted to what organisms can potentially breed with each other. Um, so, for example, you could never, under natural, normal breeding conditions, ever move a gene between a plant and an animal. It's just impossible. Um, or between a bacterium and a person or whatever. So what we do is we now have methods that allow us to do this. Um, so let's go through what needs to be done. So um, first you have to find your gene, and typically you do this by sort of working backwards. You have a protein that you know the gene is responsible for making. Um, And what you can do is, once you know which protein is that you want to make, you can basically back engineer the mRNA that this, or the potential mRNAs that this protein would be made from. And once you have the batch of mRNAs, that the potential mRNAs, you can figure out what the gene should look like, or approximately look like. Um, And so once you have that, you can then go through all this sequence data, this stuff we've talked about, um, and you can go and look for the gene and find out where it is in the genome. Um, so what you can then do is use these mRNAs as probes. And so you mix up a batch of these potential mRNAs. You've got some DNA where you think the gene is. You use restriction enzymes to cut the DNA up into the smaller, more manageable pieces. You use gel electrophoresis, as we talked about, to separate the pieces based on their size. And you can use this technique called southern blotting, and it got its name because it was sort of uh, invented, if you will, by someone whose last name was Southern. Um, and I don't want to go into the details of how this works, but what happens is you expose your gel with the DNA in it to these little pieces of mRNA, these probes. These probes are typically labeled with a radioactive material. Um, and what you do is you put film, okay, this photographic film, on top of your gel, and that's what the yellow thing is here. You have paper on top, okay? It can just be paper towels. And what happens is the solution that the gel is in gets sort of pulled up into the paper towel, wicked up, and the pieces of DNA end up um, being, well, let me back up for a second. What happens is where the probe has stuck to a piece of DNA, okay, that is a gene that matches with the mRNA essentially, it will expose the film. All right, the radiation will cause the film to be exposed, and you'll see amongst all your bands, all your pieces of DNA, you'll be able to see which ones um, are potential matches, potential genes for this protein. Then what you can do is isolate the DNA from your gel. There's ways to pull it out and begin to study it. Um, Now, you can also do what we did the other day and do blast searches to try to find um, known sequences of DNA that may contain our gene. Um, BLAST, by the way, stands for Basic Local Alignment Search Tool. It takes your sequence and essentially tries to align it up with other known sequence and tell you whether you have matches. Now, once you've found your gene, to study it and make use of it, it helps to make copies of it, or essentially to do DNA replication in a test tube. And the technique that does this is what's known as the polymerase chain reaction, or PCR. And this technique was thought up and invented by a guy named Kary Mullis, who won a Nobel Prize for that work and also cashed in big time on it because it was a pretty significant invention, patented, of course, and it's used all over the place in labs worldwide. There's probably tens to hundreds of labs over the Vanderbilt campus that use this technique probably on a daily basis. What you're doing again is DNA replication in a test tube, and the way that the technique works is essentially, you'll remember in normal DNA replication of cells, we use DNA helicase to separate the DNA. But what we use here is heat. We heat up a piece of DNA and the two strands come apart. All right. What we do is we then mix the DNA with primers that attach in places on either side of the DNA that basically straddle the gene. And this is a condensed version. This would be much longer, and your primer might be here and here. And so our gene is in this section of DNA that we want to copy. We attach primers, and then we mix in also DNA polymerase that will then, and a bunch of nucleotides, because you have to have nucleotides to make up your new piece. And the polymerase will go to where the primer is and move along the blue piece, the template piece of DNA, and make our new copies, our new pieces of DNA. We've gone from one piece to two pieces now in one cycle. Now, one of the innovations that Mullis thought of is that when you heat DNA, it has to go to a pretty high temperature. In fact, it's usually around 90 degrees Celsius, which is way beyond any temperature you would find inside of us. If you were to take our DNA polymerase, that is human DNA polymerase, and expose it to that high temperature, it would break down and it would not function properly. But what he thought of is, well, let's see, there are other organisms that live at these high temperatures. In particular, there are bacteria that live in hot springs. They have DNA polymerase just like any other living organism, and their DNA polymerase can handle these high temperatures. And so they basically extracted this DNA polymerase from these hot spring bacteria and were able to get it to work in this technique. And polymerase is polymerase. It doesn't matter where it comes from. It will build new copies of the DNA molecule. All right, so what you do is, so you sort of heat it up, you attach the primers, you mix in the polymerase and the nucleotides, you build up your new pieces of DNA, and now you have two copies. You just repeat this process again. You heat it up again, mix in the primers, polymerase, nucleotides, and you go from 2 to 4 copies. You do it again, you go from 4 to 8 copies. And you'll do this anywhere from 35 to 40 cycles. And you can perhaps try to figure out how many copies of DNA you're going to have after about 40 cycles. And we'll see what you come up with. It's a lot. Let's see. So now when you have that many copies of DNA, it gives you a lot of DNA to work with, to do your research with. It's very, very useful technique. All right, so now we've got lots of DNA. How do we move that DNA from one source to another? Okay. How do we create recombinant DNA? That is, how do we take DNA from two different species and splice them together? Well, those restriction enzymes come in handy, in particular the ones that create sticky ends, because you can have your destination DNA that you expose to the restriction enzyme, and your source DNA that you expose to restriction enzymes. The sticky ends should match up, and you'll be able to basically splice them together. Again, creating recombinant DNA. This is often done with what are called plasmids. Plasmids are extra pieces of DNA, circular pieces of DNA that bacteria and yeast have. A bacterium will have sort of its main chromosome, but then they'll have this plasmid often. And so what you can do is have a gene we have isolated from people, from humans. Let's say the gene in particular, this one is the human growth hormone gene, HGH. It codes for a protein known as human growth hormone, which promotes growth in people. So you now take this gene and you have cut it out of the chromosome, essentially using a particular restriction enzyme, in this case EcoR1, that creates these sticky ends. We then take a plasmid from a bacterium, say E. coli. We expose it to EcoR1, so it cuts the plasmid. The sticky ends from our human gene and our plasmid will match, and we can splice it in. We now have a plasmid that has this human gene in it, HGH. Through the process of what's called transformation, we can get a bacterial cell to take in that plasmid. And what will sometimes happen is the plasmid and will sometimes fuse with the main chromosome, or pieces of the plasmid will bind with the main chromosome. And so the goal is essentially to use the plasmid as a vector to move our human gene into our bacterium and into the main bacterial chromosome. All right, what will happen then? Well, essentially the bacterium is now a transgenic organism, and it will start to make human growth hormone, which is once you've got this worked out, it's a relatively cheap and abundant way to make a lot of this protein, which sometimes is used to treat people who have mutations where they don't produce enough of this human growth hormone and they're particularly short. This has also been done with insulin, the gene that makes insulin, which helps control blood sugar levels. Diabetics need extra insulin often, and so now there have been bacteria that are engineered to have this gene and make these particular proteins. Now, this transformation process is not foolproof. It's not 100% efficient. Sometimes the plasmid is taken up and sometimes it isn't. Sometimes once the plasmid is taken up and the genes and sometimes they're transferred to the main chromosome, sometimes they're not. So the question is, how do we find out which of our bacteria have successfully taken in the human gene? Well, what you do is you use a genetic marker, and often that is a gene for antibiotic resistance. Notice in our plasmid here, this is the plasmid where we'll splice in the gene for insulin or human growth hormone, for example, but it also has genes for antibiotic resistance, in this case ampicillin resistance and tetracycline. So what happens is, in addition, so the bacteria where this process has successfully happened will not only incorporate this human growth hormone gene or the insulin gene, but they will incorporate a gene for antibiotic resistance. And therefore, we can take the thousands or even millions of bacteria that we are trying to transform and use this antibiotic resistance as a way to mark or screen out those that have been successfully transformed. We basically just expose all the bacteria to the antibiotic. Those that survive, the colonies that survive, are the ones that have been successfully transformed. All right, I'm going to stop there because I'm almost out of time. We'll do the rest of section 2 in a second video.